Hello and welcome to Pathology Decoded. I am Dr. Rana, your guide through this platform that breaks down complex medical concepts into easy to grasp bites, helping students like you ace your exams. If you are here watching this video, I believe that you are looking for a way to understand and retain pathology better, and I am going to help you do just that. If you find this video helpful, please like, subscribe, and share your thoughts in the comments. Your feedback fuels future content. Now let's dive in and make pathology accessible and enjoyable. Cell injury. It is a fundamental concept in pathology serving as the cornerstone for understanding the development of pathological lesions. Mastering this concept is essential for interpreting the various pathological changes encountered in clinical practice. So what actually is cell injury? As per the definition given by Robbins, cell injury is defined as a sequence of events that a cell demonstrates when it encounters stress because of changes in its internal and external environment. Now this is quite a complex statement. Let's try to simplify it and break it down into pieces. First thing, it's a sequence of events, and where is it seen? It is seen inside a cell. And when is it seen? Whenever the cell encounters stress. So on the basis of these 3 points, we can give a very easy definition of cell injury, that cell injury is sequence of events seen in a cell when it encounters stress. A cell, when it is normally present inside the body, we say that it is in a homeostatic state. This cell, whenever it faces a change in its functional demand, it could be increase in the functional demand or it could be a decrease in the functional demand, it will undergo what we call as adaptations. These adaptations can be in form of atrophy, hypertrophy, hyperplasia, metaplasia, and dysplasia. Now all these adaptations, if the functional demand is removed and cell is again presented to its homeostatic state, it will restore itself and become a normal cell. But if this stress is of higher nature, although still mild and still moderate, the cell will undergo injury which we call as reversible cell injury, and then the cell will be present in form of degenerations, subcellular alterations, and intracellular accumulations. But again, if this mild to moderate stress is removed, the cell does not die, but instead it repairs and heals itself. If this stress is of severe nature or if it persists for a long time, then the cell undergoes what we call as irreversible cell injury. This irreversible cell injury presents in form of necrosis, apoptosis, and gangrene. These are the 3 ways in which the cell dies. So irreversible cell injury leads to cell death in 3 ways: necrosis, apoptosis, or gangrene. So what are the causes of cell injury, or what is the etiology of cell injury? In pathology, the causation of any pathology is known as etiology. So if we have to see the etiology of cell injury, we broadly divide it into 2 categories. One is the genetic causes and another is the acquired causes. The genetic causes, they include first extra chromosome. So in a condition like Down syndrome, all of you know that we have got number 21 chromosome. Instead of being present in pair, we have got it in triplets. So there are 3 sets of chromosome 21 in Down syndrome, and that leads to different type of cell injuries in the body. Secondly, we have a condition known as sickle cell anemia. Here what happens, one amino acid, the glutamic acid at position 6, is substituted with amino acid valine. Now here you see glutamic acid is a hydrophilic acid while valine is a hydrophobic acid. Because of its hydrophobic nature, Whenever the RBC is exposed to acidic environment or low oxygen environment, the hemoglobin inside it undergoes crystallization, and RBC crystallizes in a sickle shape. So here you see we have got this sickle shape RBC. So that leads to injury to the red blood cell. Now, another is enzyme defect known as G6PD deficiency or glucose 6-phosphate dehydrogenase deficiency. This again leads to injury to the RBCs. This G6PD is important to neutralize various free radicals which are formed inside the RBC, as well as it is required for energy production by the RBC through HMP shunt pathway. If this enzyme is deficient, then those individuals, their RBCs, whenever they are exposed to acidic environment or they are exposed to uh, low oxygen environment, the RBCs starts getting damaged or they get injured. Finally, we have got large number of DNA damages and mutations which we face in our life, and these leads to various changes in the cells which make the cell prone to cell injury. So this was about the genetic causes of the cell injury. Now let's come to the acquired causes of cell injury. The most common acquired cause of the cell injury is oxygen deprivation, and this is also known as hypoxia. Hypoxia is made up of two words, hypo and oxia. Hypo means less, oxia means oxygen. So any condition in which there is lack of oxygen or the less oxygen is present to the tissue, we call it as hypoxia. So what is etiology of hypoxia? The most common etiology of hypoxia is ischemia. Now, ischemia is a situation where the blood supplied to the tissue or an organ is compromised. What can cause ischemia? So here you see we have got a blood vessel, and in this blood vessel the lumen of the blood vessel is covered by plaques, yellow colored plaques. We call them as atherosclerosis. So basically this is a fat deposition in the vessel. So whenever there is deposits of the fat in the vessel, it will occlude its lumen, and because of that the blood cannot flow into the vessel and reach the tissue. So lack of blood means lack of RBC. Lack of RBC means the hemoglobin which is carrying the oxygen cannot reach the tissue, and the tissue gets deprived of the oxygen, leading to hypoxia. Now, what could be the causes of ischemia? One, I have told you, that deposit fat plaques deposits known as atherosclerosis that can lead to ischemia. Then we have got cardiorespiratory failure. Now, this cardiorespiratory failure, again, that means the heart is failing, or due to any reason, if heart is not able to pump sufficient blood into the body, the blood will not reach in adequate amounts to the tissue, and that would lead to hypoxia. Secondly, if the lungs are damaged or due to any reason lungs are not functioning properly, then the blood which reaches the lungs cannot be oxygenated properly. Now this leads to less oxygen in the hemoglobin, and the tissue is deprived of this oxygen, leading to hypoxia. Now there is one question which is commonly asked in the Viva regarding this, whether there can be hypoxia without ischemia. This is hypoxia versus ischemia. Ischemia, like I told you, there is lack of blood supply to the tissue. Hypoxia is lack of oxygen to the blood tissue. Definitely, ischemia will always cause hypoxia. But the question is, can there be hypoxia without ischemia? Yes, it is possible that the blood reaching the tissue is in sufficient amount, but still the tissue is not able to get the oxygen. Why it happens? Now, this can happen in a condition called as severe anemia. Now, in severe anemia, what has happened is that the number of RBCs or the RBCs which contain hemoglobin is decreased. So, low amount of hemoglobin means that low amount of oxygen will be carried by the blood to the tissue. And if the demand of the tissue is high, it will be getting less oxygen. So, the tissue will suffer from hypoxia, although the blood which is reaching the tissue is in sufficient amount. Then we have got another condition which is known as carbon monoxide poisoning. Now this carbon monoxide is produced whenever there is burning with like wood or coal, etc. Whenever these are burnt, carbon monoxide is produced. Now carbon monoxide has got a special ability by which it is able to bind irreversibly to the RBCs. It basically binds to the hemoglobin, and this irreversible binding to the hemoglobin leaves no place for oxygen to bind to the hemoglobin. So these RBCs, they are not able to carry the oxygen. So although the blood is reaching the tissue in sufficient amount, still the oxygen is not reaching there. So the tissue will suffer from hypoxia, although there will be no ischemia. So yes, there can be hypoxia without ischemia, and the conditions are severe anemia and carbon monoxide poisoning. The second group of acquired causes of cell injuries include physical agents. Amongst these, the most common one is the mechanical trauma. And this mechanical trauma can be caused by roadside accidents, falls, or injuries like crush injuries and penetrating wounds, etc. Then we have burns and deep colds. Burns could be due to fire or electrical burns, while deep cold can cause tissue injury by causing frost bites. The third group includes sudden changes in the atmospheric pressure. These sudden changes in the atmospheric pressure can cause a condition known as barotrauma. This is seen in the mountain climbers who go to a very high altitude at a very fast rate, and they are exposed to low atmospheric pressures. This is also seen amongst the deep sea divers who dive fast and they are suddenly exposed to very high atmospheric pressure. When they come up, they suffer from a disease known as Casson's disease. Lastly, amongst the physical agents, we have radiations. Radiation injuries occurs when tissues are damaged by exposure to the ionizing radiation. One common example is a Chernobyl nuclear disaster. Also, the exposure of our skin to ultraviolet radiations or exposure to X-rays, repeated exposure to X-rays, etc., can also lead to cell injury. The third group of acquired causes of cell injury include chemical agents and drugs. Amongst these, glucose and saline, they are very commonly used in the patient who are hospitalized. High concentration of glucose or high concentration of saline, known as hypertonic saline, can cause cell injury by dehydrating them or damaging them. Then exposure to arsenic, cyanide or mercury can also lead to cell injury. Arsenic is seen in mining and smelting industries, and exposure there can lead to arsenic poisoning. It is also present in pesticides and herbicides. Cyanide is seen in gold and silver extraction industries. Cyanide exposure can also occur if there is heavy consumption of bitter almonds. Mercury is seen in patient consuming high amount of seafood. Mercury is also present in dental amalgam and can lead to cell injury. Alcohol in high levels can lead to cell injury, and the organs which are vulnerable to alcohol poisoning are liver and our nervous system. They can get badly damaged or injured by the alcohol exposure. Then air pollution along with insecticides and herbicides, these all are environmental chemical agents which can lead to cell injury. In fourth group, we have infectious agents, and these infectious agents include various bacteria, viruses, fungi, and parasites. All of them are causing cell injury in our body on a daily basis. Got immunological reactions, and these immunological reactions or immunity involved conditions, they include external agents and self antigens. External agents are the agents which are foreign bodies against which the body mounts a immune attack, and during this attack the cells of the body, they are injured. On the other hand, self antigens are involved in autoimmune diseases. Autoimmune diseases occur when the immune system mistakenly attacks the body's own tissues, thinking that they are foreign invaders. So the self cells of the body, they are taken as foreign particles, and this can lead to the inflammation and damage to various parts of the body. Now, amongst the autoimmune diseases, common ones are rheumatoid arthritis. This is a condition in which antibodies are formed against the synovial lining of the joints of the body, and the patient suffer with joint pains. Another category is type 1 diabetes, in which there are antibodies against the insulin producing cells of the pancreas. Now they are damaged, and so no more insulin is secreted, and the patient suffers with the insulin dependent diabetes. And third common condition is a SLE. Now SLE stands for systemic lupus erythematosus. Now this is a condition in which there is an autoimmune response by the body against the multiple organs. The name of the disease is systemic because whole of the body and multiple organs are involved. And lupus erythematosus, erythematosus means that there are red color rashes over the body, and lupus is a Latin word for wolf. Those rashes look like as the wolf has taken a bite in the skin. So the word systemic lupus erythematosus means it's a disease involving multiple organs with red colored rashes on the body, which are uh, looking like as if the wolf has taken a bite from it. We have got various nutritional imbalances which can lead to cell injury. Amongst this, common one is protein calorie deficiency. Now, protein and calorie deficiency, this can lead to nutritional disorders. If there is any protein disturbance, then the child presents with a bloated body, and this condition is called as kwashiorkor. On the other hand, if protein deficiency is associated with calorie malnutrition, then the child presents with massive muscle and fat loss, and this condition is called as marasmus. So in both these conditions, there is severe cell injury. Then various vitamin and mineral deficiencies can lead to cell injury. One very common example is, a condition known as scurvy, which is caused due to vitamin C deficiency. So there is bleeding and there is damage to the superficial skin and the collagen of the body. And then excessive or excess nutritional imbalances can also lead to cell injury. These include obesity, and excess cholesterol intake can also lead to deposition of the fat in the blood vessels in a condition known as atherosclerosis. Due to various acquired causes or genetic causes, the cell injury which is caused in the cells has got four major consequences. The first one amongst them is disruption of the cell membrane integrity. The cell membrane of the cell is made of phospholipid bilayer, and when it gets disrupted, the cell faces electrolyte imbalance along with loss of various channels and pumps which are present in this membrane. At number 2, we have got decreased aerobic respiration. That means the oxidative phosphorylation suffers and there is decrease in the aerobic respiration. This decreased aerobic respiration will lead to less production of ATP by the cell, and the cell will not have sufficient injury to survive on itself. This leads to the cell damage. Third category is decreased protein synthesis. Now this has got consequences like the cell won't be able to divide because protein synthesis and new organelles formation is required for the cell division. At the same time, by decreasing the protein synthesis, the various functional proteins inside the cells, examples include like coagulation factors, various enzymes or hormones, etc., they won't be synthesized in the sufficient amount. This leads to compounding of the cell injury. And finally, the fourth attack by these agents on the cell is impairment of the genetic apparatus integrity. Now this causes the DNA damage. This DNA damage, it is largely due to deamination of the cytosine residues to the uracil residues. And whenever the DNA damage occurs, there are certain genes in the body like P53 which get activated, and they give the signal to the DNA repair genes inside the body to repair the DNA. If that damage is repairable, these genes will try their best to repair the DNA. But if due to certain reason or due to too much of damage they are not able to repair the DNA, then what happens is the other set of genes that is kicked in, which causes the cell to undergo apoptosis, and the cell dies by committing the suicide. We will take that up when we will be discussing apoptosis. So these four consequences of the cell injury, they are universally seen whenever the cell is exposed to all those agents which were acquired or genetic, which I talked about in the previous slides. Cell function is lost before the cell death, and the morphological changes are last to appear. What this means is that the cell function will be impaired initially and the cell would still look normal. But if the stress persists, then slowly and slowly the changes will appear in the cells, which we will be able to see in the microscope. Whenever the cell is exposed to the stress caused by various genetic or acquired agents, the injury begins in a reversible manner and it is called as reversible injury. Taking the model of hypoxia, let us explain what happens to the functions of the cells and how these functions are affected and what morphological changes occur inside the cell when there is loss of those functions. So the first thing to occur is ischemia. Now this ischemia will lead to hypoxia, and the first thing which this hypoxia will cause is decrease in oxidative phosphorylation. Now all of you remember that oxidative phosphorylation involves burning of one molecule of glucose and production of ATP by the mitochondria. So this decreased oxidative phosphorylation will cause decrease in production of the ATP. All of you know that molecule of glucose, if it is undergoing aerobic respiration, then one molecule of glucose produces 38 ATPs. On the other hand, if it is undergoing anaerobic respiration, then the molecule of glucose will be producing just 2 ATPs. So there is a difference of 19 times. So in the absence of oxygen, decreased oxidative phosphorylation will turn the aerobic respiration into anaerobic respiration, and this decrease in the ATP will affect one very important thing, and that is sodium pump. The sodium pump which is present in the cell membrane. The effect on this sodium pump is responsible for all the changes which we see in the reversible injury. To understand the effect of sodium pump functions disruption leading to reversible injury, let's see this model of the cell. Here I have made a model of a cell with the cell membrane. Inside the cell there is nucleus and there is present this mitochondria. This area is called as intracellular region, while this area is called as extracellular region. Now, normally you know that in the intracellular region, large amount of potassium is present. So these are the potassium molecules represented by the K. While outside the cell in the extracellular region, large amount of sodium molecules are present. So sodium is more in the extracellular region while potassium is more in the intracellular region. This is the normal function of the cell. Now what happens that since potassium is more inside and less outside, due to osmosis, because the plasma membrane of the cell is a semi-permeable membrane, this potassium will start diffusing outside. This is a passive diffusion. This is bound to happen, and at the same time again due to osmosis these sodium molecules they will start moving inside the cell. Again this is a passive diffusion. This keeps on happening. Potassium goes out while the sodium goes inside the cell, because the plasma membrane is a semi-permeable membrane, and this is the normal thing which we expect to see in a diffusion. Now if this continues, what is going to happen is that after a certain time sodium levels will start increasing inside the cell, because how they have come by the passive diffusion, while these potassium molecules they will come outside of the cell. Now when they are coming outside of the cell, these potassium molecules they will accumulate in the outside, and the sodium will start accumulating in the inside the cell. Now this is not a good thing. So ultimately, to come back to its normal state or the homeostatic state, cell has to do something, that the sodium which has leaked inside should go outside and the potassium which has leaked outside should go inside. Now to do this, the cell in its plasma membrane has got something called as sodium potassium pump. This sodium potassium pump, it gets activated, and when it gets activated, what it will do is that it will start pushing these sodiums outside. So this sodium stated by N, let's say we have got this N, and since it is going out, let's say it is O. So we have got two words N and O. At the same time, this pump will also help potassium molecules to come inside the cell. So potassium molecule K, and then it is going in, so let's say it is with the I, and to do this function this pump needs the energy, because it will be doing against the concentration gradient. Remember I told you sodium is more outside. Now from inside, if it has to push the sodium outside, it has to utilize energy, and that energy is provided by the ATP. Same way, potassium is more inside the cell. So to push even more potassium from outside into the cell, this pump will need energy. That's why it is a pump. It is working against the concentration gradient. So now what you have got? We have got Nokia. So remember from this Nokia that N for sodium, O for out. So sodium goes out. K for potassium, I for in. Potassium comes in. How? By the sodium potassium pump which uses A, which is ATP. Now again, one molecule of ATP utilizes one molecule of ATP is utilized by the sodium pump to push two molecules of potassium inside and three molecules of sodium outside. So here we have got a very good mnemonic to remember. Just remember Nokia and 1, 2, 3 in the opposite direction. Sodium out, potassium in with the utilization of ATP. And the number is 1 ATP molecule to push 2 potassium inside and 3 sodium outside. Now, so this pump goes on working, this pump goes on working. The all that passive diffusion which is occurring, which is pushing the sodium in and the potassium out, the pump will work and it will again bring the cell into its normal homeostatic state by pushing 2 potassium inside and 3 sodium outside. But if this pump gets damaged or if there is a cell membrane damage, what happens? Over the time, what is going to happen is that sodium will start accumulating inside the cell. So more sodium will be present inside the cell and more potassium will be present outside the cell. Why this is happening? Because due to hypoxia, the membrane got damaged. The membrane got damaged, the pump got compromised. At the same time, since there was decreased oxidative phosphorylation, this mitochondria is not able to produce sufficient amount of ATP. So the pump cannot use this ATP. In this situation, what is happening is that sodium will stay inside the cell and potassium will stay outside the cell. To remember it, we have this thing, sodium stays and potassium flees. Now since the potassium is coming outside, and also we know that potassium is more in the intracellular region, if this membrane gets damaged, if this cell gets injured, whole of this potassium is going to come outside. Now, if we take the sample of such a patient, if we take the blood sample of such a patient, we will see that the potassium level in the serum will be high, and this is a condition called as hyperkalemia. So all the patients who are suffering from hypoxia, who are lying in the ICU or in the hospital, if their sample is taken and potassium is coming high, the doctors come to realize that oxidative phosphorylation is being affected and the cell damage is occurring, because of which potassium from inside is coming outside in the blood. Now, coming back to this, so decrease in the sodium pump is going to cause what? These three things. One, there will be influx of the sodium, and since the sodium is more inside, the water will follow. This will lead to swelling of the cell. Second, potassium will go out, and third, the calcium will also come inside the cell. Now this is a important thing because calcium is a responsible molecule for activation of various enzymes inside the cell. This leads to cellular swelling, loss of microvilli, formation of blebs, and endoplasmic reticulum swelling. Okay, on the other hand, what is happening is that there is increased glycolysis. This increased glycolysis is because the oxidative phosphorylation has become anaerobic. Now this anaerobic will lead to glucose being converted to lactic acid. This lactic acid accumulates inside the cell, and this is responsible for decrease in pH. Also, more glucose will be required because now only 2 ATP molecules are being formed instead of the 38. So this glucose is going to come from the glycogen chains. So glycogen deposits, they will start disappearing in the body. Now this will lead to increased acidic environment, will lead to clumping of the nuclear chromatin. Also there are other effects which involve detachment of the ribosomes from the endoplasmic reticulum, and all of you know that ribosomes are responsible for the synthesis of protein in the cytoplasm. So there will be decreased protein synthesis. Now decreased protein synthesis along with acidic environment, along with the damage to the phospholipids in the membrane, there will occur deposition of the lipid and their breakdown products inside the cell, which will lead to lipid deposition. So if we have to see diagrammatically, we have here got a normal cell which has got a nucleus and which has got here the mitochondria, endoplasmic reticulum, and the cytoplasmic vacuoles. Now whenever there occurs this reversible injury, one thing, the cell becomes bloated, it becomes swollen, and the membrane starts outpouching outside. These are called as blebs. So first of all, there is cellular swelling and microvilli that gets damaged, and there is formation of the blebs. Then in the mitochondria, there appears small amorphous densities. Now you have to remember these words. One thing, these densities, they are not large, they are small, they are amorphous, and so that means amorphous means they are homogeneous. They are not coarse, they are homogeneous small densities in the mitochondria. Then endoplasmic reticulum, it gets swollen, and the ribosomes which are attached on the endoplasmic reticulum, they are dispersed. They get separated from the endoplasmic reticulum. Then we have got autophagy by the lysosomes. The lysosomes invite the cells, those mitochondria which are getting damaged, it will start eating them up. Other cell organelles also, they are got phagocytized by them. So this is called as autophagy. Auto means same and phagy means eating. The cell starts eating itself. And finally, we have got clumping of the nuclear chromatin. So the nuclear material inside the nucleus, this DNA, that starts getting clumped, and it starts appearing as a dark patches in the microscopes. So these are the changes which we see whenever the cell is undergoing reversible cell injury. Now, if we see it under the microscope, you see blebs and loss of microvilli and small amorphous densities, endoplasmic reticulum swellings. They are very difficult to appreciate under a compound light microscope which is present in the lab. They can be appreciated by looking the cell under electron microscopes, but electron microscope is rarely available in the labs. It's seen in a very high-end labs. So when a pathologist sees the cell under the compound microscope, he is able to appreciate two things. One, that the cell appears swollen because the water has accumulated inside the cell. So that is called as hydropic change. And the vacuoles start appearing in the cytoplasm of the cell. Because those lysosomes, they become enlarged and they start doing autophagy, so they appear as cytoplasmic vacuoles. So two things which the pathologist is able to see is water inside the cell causing its swelling and large amount of vacuoles in the cytoplasm. So these changes are also commonly called as hydropic change or the vacuolar degeneration. So now remember that in reversible cell injury we have got hydropic change and vacuolar degeneration. Sometimes in the exam, straight away asking a question, write a note on reversible cell injury. The question comes, briefly discuss hydropic change or briefly discuss vacuolar degeneration. Please don't get confused. It's just you have to explain whatever I have told in these slides regarding the reversible cell injury. Now, like I told you in the first slide, if the stress becomes severe or if it persists for a long time, ultimately cell will go into a state known as irreversible cell injury. Now, since it cannot be irreversed, this injury will go on progressing until the cell dies, and that is known as cell death. Now here what happens? First of all, there is membrane injury. Now this injury to the membrane, it will lead to loss of phospholipids which are present in the membrane, cytoskeletal alterations, so the damage to the membrane and shape of the cell that will get damaged, and this will lead to formation of the myelin figures. Then there will be generation of the free radicals and lipid breakdown and other things, they are going to occur inside the cell. Now due to this damage by the membrane, what is going to happen is that enzymes which are present in the cells, they will start escaping in the blood. So depending upon which cell is being injured, we can measure the level of these enzymes in the blood, and we can say that this particular tissue or this particular cell of the body is getting injured. For example, if we do the test for creatine kinase or lactate dehydrogenase or cardiac troponin and we find that the level of this enzyme is high, we can very safely say that it's the heart muscle which is getting injured. So patients who are suffering from heart attack or myocardial infarction, in those patients, if we check the level of these enzymes, we can tell how much damage to the heart has occurred. That means how many cells of the cardiac muscles are damaged. Then if the damage is to the pancreas, etc., then there will be increase in lipase and amylase. If the damage is to the hepatocytes or the liver cells, then there will be increase in SGOT or SGPT. So this is one important practical aspect, that why the level of the enzymes increase and how by knowing which enzyme has increased, a clinician can tell that which organ cells are getting damaged. The second part is that when the membrane gets injured, the calcium which is present in the blood, it starts coming inside the cell. This calcium, it reaches in the mitochondria and it leads to mitochondrial damage. All of you know that mitochondria is involved in oxidative phosphorylation. That means production of ATP. So mitochondria are the storehouse of the energy. They are the electrical centers where the energy is produced. So they produce ATP, and if they gets damaged, this ATP production will stop. No energy, the cell will not be able to survive. One thing. The second thing is that this calcium, along with the decreased pH, when it comes inside the cell, it will lead to release of these activation of the lysosomal enzymes like phospholipases and proteases. As the name suggests, proteases will digest the proteins and phospholipase will digest the phospholipids which are present in the membrane. So calcium will activate these lysosomal enzymes, and calcium also acts as a base for the reactions to occur which will lead to damage of these. Now this will lead to protein digestion in the cell, and this will lead to nuclear changes. If we look at the normal cell, the nucleus of that cell contains acidic material. What acidic material? RNA and DNA. Remember, these are acids, ribonucleic acids and deoxyribonucleic acids. Now, if you remember that if they are the acidic material, they will have a philia for basic dye. That means they will have affinity to bind with the basic dye. If we stain the cell, the basic component of the dye will bind with the RNA and DNA, and that's why nucleus of the cell appears blue in color, because basic dyes are blue in color. But if there is DNA damage, if there is RNA damage, the acid inside the nucleus that will decrease, and due to that the basic dye will not be able to bind, and so we say that the basophilia has decreased. So instead of cell nucleus appearing bluish in color, it will now appear less blue in color, and this is called as decreased basophilia. So here we had a normal cell and it had gone reversible injury. Now let's see, when it becomes irreversible injury, what happens. The one thing to happen is that there is disruption of the membrane. Now this disrupted membrane causes enzymes etc. to leak outside, calcium to go inside, and along with that there is damage to the structure of the cell. Secondly, we have got lysis of endoplasmic reticulum. So the endoplasmic reticulum, which had earlier been just swollen with the detachment of the RBC, now they will start getting dissoluted, they will start getting damaged. In the mitochondria, we will have large coarse densities. Remember, in reversible, we had small amorphous densities, but here these densities, due to calcium coming inside the mitochondria and getting deposited, it will appear as a large coarse densities. And finally, there will be rupture of the lysosomes, and the rupture of these lysosomes will release the enzymes, phospholipases and proteases, DNases, collagenases. All these enzymes will be released, and in the presence of calcium, they will become very aggravated. They will become activated and they will start doing their digestive functions to digest the collagen, to digest the phospholipids, to digest the protein material inside the cell. And this will be followed by nuclear degeneration. Now the nucleus of the cell that will start getting damaged, which will lead to decreased basophilia. Thus membrane of the cells, now what is going to happen, that the proteins which are present in the membrane of the cells, when the water is entering inside the cell, it will start getting fixed between the layers of the membrane, and the membrane it appears lamellated. So if we see it under the microscope, electron microscope, mind you, they appear as the myelin figures. Now these myelin figures, they are similar to the myelin sheath of the nerves, but they are in no way related to them, but they are like those myelin layers. Now this nucleus, when we were talking about this nuclear degeneration, this nucleus can get damaged in three manner. One, all the material in the nucleus, nuclear material, that can become clumped into a small glob. We call that as pycnosis. Then that nuclear material, it can all get dissolved. So if you look it under the microscope, you will not be able to see any nuclear material. It will appear as the nucleus has become filled up with some fluid. There is no solid particle, no DNA, chromosomes, etc., which you can visualize. Then it is called as karyolysis. And finally, that nuclear material, it can also get fragmented into small particles. Then we call it as karyorexis. So these are three ways in which nucleus of the cell dies, and they are important. They are important from the bio point of view also. So one, the nuclear material can become condensed into a single glob. We call it as pycnosis. It can become all dissolved and disappear. We call it as karyolysis. Or it can break down and get fragmented into very small particles. Then we call it as karyorexis. Okay, finally now come to the differences between reversible cell injury and irreversible cell injuries. We have talked about both. We have talked about the changes in their functions. We have talked about various morphological pictures which we have seen in both. Now let's compare them. Number one, in reversible cell injury, there is cellular swelling with small amorphous densities in mitochondria, while in irreversible cell injury, there are large coarse densities in the mitochondria. In the reversible injury, there are loss of microvilli, while in irreversible, we have got membrane disruption. Then in reversible cell injury, there are formation of blebs, and in irreversible cell injury, we have got damage to the cell membrane with the formation of myelin figures. In the reversible cell injury, endoplasmic reticulum undergoes swelling and the ribosomes get dispersed or detached from it. While in irreversible cell injury, whole of that endoplasmic reticulum simply gets dissolved, dissoluted or disintegrated, which we call as lysis of endoplasmic reticulum. Then in reversible cell injury, the lysosome starts autophagy. That means their structure is intact and they are trying to eat the organelles inside the cell which are no more required. But in the irreversible cell injuries, these lysosomes themselves are damaged and they get ruptured, releasing the various enzymes inside the cell, causing dissolution of the other parts of the and other components of the cell. And finally, in reversible cell injury, there is just clumping of the nuclear chromatin. While in irreversible cell injury, the nucleus gets totally degenerated and it can undergo degeneration in three forms. First one was pycnosis, then karyolysis or karyorexis. So these are the common differences which can be visualized in the reversible and irreversible cell injury in electron or in the compound microscope. So that finishes with the part of reversible and irreversible cell injury. So please like and subscribe to my channel and share it with others. And if you find video helpful, please share your thoughts in the comments. Thank you everybody.